We present SCANViz, a visual analytics system for deep learning experts to interpret, explore, and interactively train the simple concept association networks. SCANViz contains three main visualization views. The first view provides an overview to different types of training statistics. It also enables users to explore the training images with different visual concepts. The second view demonstrates the network architecture of the beta VAE and SCAN. Through the interface, users can flexibly switch between different architecture modes to change the data flow. Meanwhile, this view also presents numerous sample reconstructions and their distributions to disclose the generative power of SCAN. The last view uses a parallel coordinates plot to visualize the latent space of the beta VAE and SCAN. The current view demonstrates a latent space with 32 latent dimensions. The light yellow and light blue bands visualize the latent representations of the input simple concept pair from SCAN and beta VAE respectively. We now demonstrate the case studies presented in our paper with the DeepMind Lab dataset. From the overview of the training statistics, it is very obvious that the reconstruction loss in purple converges much slower than the two latent losses in orange and green. The observation leads to the hypothesis that SCAN's decoder is more difficult to be trained than its encoder, which has been explained in our paper. The image-to-symbol curve converges much slower than the symbol-to-image curve, which also verifies the hypothesis. The variance curves for the 32 latent dimensions of SCAN also converge very quickly. Only 7 out of the 32 curves have values greater than 0, which are the dimensions that really capture the visual concepts of the images. The SCAN view allows users to investigate different data paths of the joint architecture of beta VAE and SCAN in different training stages. For example, we now switch to epoch 10 and explore the symbol-to-symbol path. The symbol of interest has gray object color, yellow wall color, gray floor color, and the object ID is ice lolly, represented by the red circle. From the reconstructed symbol, we can see the wall color is incorrectly reconstructed as red, and not a single value is dominating the reconstruction result. This can also be observed from the distribution of the 60 reconstructed samples. The incorrect reconstruction is majorly due to the poor quality of the SCAN's decoder at this stage. If we move to a later stage, the reconstruction becomes correct, and gradually, we can see SCAN becomes more and more confident in reconstructing the yellow wall color. Back to epoch 10. Although SCAN cannot reconstruct the yellow wall color, it performs very well when reconstructing the symbol instance with red wall, indicating a different difficulty level in reconstructing different symbol instances. Coming to the symbol-to-image mode with the instance of yellow wall, we can see the generated images are mostly with a yellow wall color. The bad symbol-to-symbol quality and the good symbol-to-image performance further verify the SCAN's decoder is not good enough at this stage. Switching to the image-to-image mode, we can investigate the quality of the pre-trained beta VAE. The forward analysis allows users to directly interact with different latent dimensions and watch how they affect the reconstructed image. For example, D4 controls the wall color of the image. D7 has no effect to the reconstructed image. D31 influences the object ID. D18 controls the floor color. D20 also controls the floor color. Note that multiple dimensions can control the same concept. To find all dimensions that affect the floor color, we switch to the backward analysis. First, we select images that differ in the floor color dimension only and visualize their aggregated mean curves in the PCP. From the red band, we find that D11, D21, and D31 also have different levels of influence to the floor color. SCANViz also allows its users to interactively train the SCAN model. First, we initialize SCAN by resetting its model parameters. The target concept to learn in this example is gray floor, which can be specified using the color circles. The circles with a cross indicate dimensions with unspecified values. Next, we select images with a target concept for training. In the first training stage, we select gray floor images with difference in object color only and train SCAN for 2000 epochs. Over the training, we can directly see the change of the 3 loss curves and the 32 variance curves. The variance curve for D20, D31, D18, and D4 have obvious increasing trend. These 4 dimensions majorly control the wall color, floor color, and object ID. Their increase means the SCAN model believes gray floor images should have restrictions on all of these 3 concepts, which is not necessarily true. Checking the generated images through symbol-to-image inference with a target concept, we see that the current scan can generate gray floor images with diversity in object color only. For the second training stage, we add gray floor images with varying wall colors into the training set. From the variance curves, the most obvious change is the drop of D4, which is associated with the wall color. Its decrease indicates the model relaxed its restraints on the wall color dimension and could generate gray floor images with diverse wall colors. To the third training stage, we add images with different object IDs into the training sets and continue to train the model for another 2000 epochs. This time, D31, which majorly controls the object ID, presents a sharp drop, indicating our scan model also relaxes constraint on the object ID dimension. Checking the generated images, we can see that the model clearly captured the target concept and it can generate gray floor images with diversity on all the other 3 dimensions. The curves for D18 and D20 kept high, which reflects the consistently strong constraint on the floor color. We have also explored the cartoon set images with SCANViz. The detailed explorations of this dataset can be found in our paper. Thanks for watching.